we're very excited here today to have Siddhartha Sharma, PhD, join us from the Titan Laboratory at Scripps Research Scripps Research period in La Jolla, California. And his talk today is leveraging circulating insulin-specific CD4 cells to measure autoimmunity in type 1 diabetes. We had a lot of interest in this talk and some international interest. So if people are unable to join us right now, we are recording this and we'll house it on our website. I know a lot of people will be watching it later as well. And I just wanted to have you say a few words, Siddhartha, about your background and where you are right now.
Okay. Well, first, thanks for having me and giving me the opportunity to present our work. I am currently in the Taint Lab at Scripps Research. I also did my PhD here, and it was focused on translational immunology, really trying to understand, can we go back from mouse models to human studies and try to better understand type 1 diabetes utilizing both. Before that, I did my master's at Boston University and also did my bachelor's at Boston University. And as far as where I'm originally from, I'm a bit of a nomad, but I was born in Illinois, but raised in upstate New York.
Fantastic. Well, you have picked the right state for better weather from Boston. So
I'd agree.
Okay, so yeah, so take it away.
Great. So today I'll be sharing the work that we recently published in Science Translational Medicine and had the opportunity to share with the community in Paris earlier this year, May this year. Title of my talk is really, we're going to try to leverage circulating insulin specific CD4 T cells as a way to measure autoimmunity. And while we have a means to track autoimmunity through autoantibodies, and these things autoantibodies have been very important for tracking autoimmunity in people at risk for type 1. And this this graph shows how much the risk increases from just transitioning from one autoantibody to two. And this has been really critical for patient care and helping prevent diabetic ketoacidosis. The one probably point that I would like to make, though, is on the x-axis. And one thing that I think autoantibodies, maybe one weakness of them, is that they measure probability of diabetes over decades. It doesn't really give an individual a good gauge of their own state. Where do they stand in the process of moving towards type 1 diabetes? Many people do become type 1 diabetic, but some don't. Another way to think about this is kind of go back to the GWAS study. So, what is type 1 diabetes, the greatest risk for type 1 diabetes? And what we know is that the MHC class 2 is the greatest contributor to the genetic risk for type 1 diabetes, and the MHC locus. And that's just shown here in this figure, sibling relative risk. The highest ratio is for the MHC locus. More specifically, though, there's a point mutation in the DQ beta 1 chain in DQ8 and DQ2 haplotypes. That's where that risk, a majority of that risk comes from. There is risk from other areas, but that's where the majority comes from. So what is the consequence of that point mutation? Well, in the Taint Lab, we spent, people before me and many researchers before me have spent a lot of time thinking about this and doing experiments. And one of the consequences of that point mutation is that there's a positive charge that's left on the MHC surface. And that positive charge can be, is on the MHC surface, and the MHC interacts with the CD4 T cell via its TCR. Previous people in the Taint Lab have proposed a P9 switch model for TCR recognition. One of the features of this is that positive charge on the MHC class 2 surface can be neutralized by a peptide loaded in the MHC class 2 binding pocket by a negatively charged amino acid in the position 9. However, it also cannot be, it may not be neutralized by a peptide in position 9. There's some flexibility there. So one of the consequences of that is that if there is a peptide that is loaded into the MHC that has not neutralized that positive charge, the second feature of this P9 switch model comes up, which is that there's negative charge residue in the CDR3 beta region. This has been shown by other people in the Dayton lab. One other consequence is that the way this P9 switch model, the TCR recognition of this MHC gives preferential activation and early expansion of T cells that recognize peptide MHCs with non-negatively charged residues. And building on all that work that's been done before, not just in our lab, but in other labs, in 2019, we published a paper in Science Immunology where we looked at the role of the position beta 57, that positive charge that remains there. What is that role for susceptibility of type 1 in a non-mouse model? And one of our findings was that if you modify that P9 peptide, what you find is that, and here's what we are showing, here's an IL-2 assay. So you have IL-2 release on the Y, and this is plates coated with IgG7 monomers. If you plate this and then put cells on isolated from the islets of non-mice, what you find is that if you have a negative charge in that peptide, so for example, in insulin 1220 here, you have a negative charge there, you can actually ablate recognition. Now, if you also look at it from T cells that are isolated from immunization, you also find the same feature. You have a ablation of recognition because you have a negative charge in that 9th position. Let's flip it the other way, though. Let's say you, clone 2, there was a negative charge in the CDR3 beta region. Let's say you change that to a glycine. What you find here is also by IL-2 assay, you can ablate recognition. And finally, by looking at SPR and affinity measurements, 2D10 is a TCR that has a negative charge in CDR3 beta. If you change that to a glycine, you can reduce affinity drastically. So this whole model is called is the P9 switch mode of TCR recognition, and what it provides is a testable possible mechanism for type 1 diabetes. What it also provides is a TCR phenotype, and this is something that we can then use not just in a non-mouse model, but also in human studies. Excuse me. And this is sort of the basis of the work for this paper, which was, can we utilize, after looking at non-mouse models and building on work from our lab, but also previous other labs over decades, can we get another way to gauge, get a more granular measure of autoimmunity? So as I mentioned, we have autoantibodies and they are excellent. They are very valuable. But the other method we have for tracking autoimmunity is just looking at beta cell function via these methods here to the left. What we're proposing is, or what we are suggesting, that maybe we can use T cells, which, given the contribution of the MHC class 2 locus to the risk for type 1, looking at CD4 T cells could be a more granular measure of autoimmunity. And that's not just something that we're proposed. This paper in Science Translational Medicine from 2017 also looked at CD4 T cells. So that's what the theme of the hypothesis here is that, circulating insulin-specific CD4 T cells can be utilized as biomarkers for anti-beta cell autoimmunity. And the way we've done this is, since it's a translational project, we have mouse models, the non-NOD mouse model, and we also have human studies. And one of the way this paper was structured, we started off with mouse work, and then we transitioned to human work. And so I'll begin with that, figure 1 of that paper. So what we first started off is we have IgG7 monomers that we've made in-house, and we've loaded them with different regions of insulin 1220 and 1321, but also loaded it with a BDC 2.5 antigen. We drew blood from non-mice biweekly between 4 and 25 weeks, and we measured glycemia. And at the same time, called the blood, stained it with our IgG7 tetramers and an assortment of antibodies, and we profiled the total PBMC population by spectral flow cytometry. What we then had to do was then take, all the measurements from weeks 4 to 25 and, normalize it, collapse it into one analysis, overlaid clustering, and then we could see, at a bulk CD4 T cell level, what was the activation state of the mice, but also at a more granular level, what is the activation state of the insulin-specific CD4 T cells. So this is the results of compiling between weeks 4 and 25. So here in the top left, we have a UMAP, and in this UMAP is all the CD4 T cells that we accumulated across all the mice. What the coloring is the different clusters that we found, and we projected that onto the UMAP. The UMAP below here is the tetramer-specific cells. So it's the same UMAP as the one above, but we just grayed out the clustering and overlaid where the tetramer-specific cells were. And we have almost 2,700 antigen-specific cells in this analysis. What we find is that most of them are here on the left, and most of the positive cells are also here at the bottom. So clusters A and B, if we go back to the top UMAP, are the non-activated cells, and mostly based off of CD62L and 44. So very low 44, high 62L, and very little expression of other activation markers like ICOS. This contrasts clusters C and D, which have higher CD44, lower 62L, higher ICOS, and as well as some PD-1. So now that we have this sort of distinction at bulk level, and we can categorize all the bulk CD4 T cell population and then project onto it where are those insulin-specific cells, we can now do an analysis with studying over time where those cells, the insulin-specific cells, are in the bigger CD4 population, but we can also compare it to glycemia measurements. So when we look at the glycemia measurements for all the mice, what we find is between 4 and 8 weeks, there's some degree of stability, though it is a slight increase. We find a larger increase in glycemia measurement at week 10, which slightly declines, but then again peaks back at week 23. Many of the mice did become diabetic, but some of them did not. However, we can also now look at the frequency and activation ratio. So when I say activation ratio, what I mean is of the tetramers specific cells, what percent of them are in the activated clusters over the non-activated clusters. When we look at that, what we find is starting at here at the top, the BDC 2.5 antigen, in terms of frequency, but also activation ratio, it's pretty much flat. There's not much change. There's a little bit of a peak and valley here towards the end, but in terms of activation, it's pretty minimal. Now, when we go to insulin 1321, what we find is in terms of frequency, we start off at one of the high points. It dips down, more or less stays flat, but then peaks again at week 25 and then declines. It's sort of inverse, though, for the activation ratio, where we find the activation's highest at week 21, and it's mostly flat otherwise. This contrasted insulin 1220. So this is insulin 1220 is the one is the peptide that does not have the negative charge in the position 9. What we find with the frequency is that for the most, it starts off stable, dips at around week 10, which corresponds to a slight increase in glycemia in the mice, plateaus again, and then declines as far as frequency. But I think where it's most interesting is that the activation ratio, where you have a small blip where there's more activated cells at week 8, which precedes the increase in glycemia, a plateauing, and then again, another increase at week 21, which precedes the increase in glycemia at week 23, and then it declines. What this tells us is that, or what this told us was that the CD4 T cells, insulin-specific CD4 T cells, could be used as a way to kind of catch fluctuations of activation and changes in frequency that could precede changes in glycemia. Now, I did mention that many of the mice became diabetic, but some did not. So what we did for those non-diabetic mice is we took them, sacked them, removed their pancreas, and looked at could we still find signatures of autoimmunity despite the fact that they did not become diabetic? And in fact, we did. So here in the left in green is a signal from insulin, and the middle panel is glucagon, and the right is the overlay. What we find is that we can find islets that are intact for insulin expression, but we have also found islets where the beta cells have been destroyed. And these are all in mice that were not diabetic. And what this shows us that just because the mouse didn't become diabetic, it did have at least bouts of autoimmunity that removed beta cells. So transitioning now to the human studies. We, between the work we did in Science and Knowledge paper and just now, we showed that it is viable to utilize circulating CD4, antigen-specific CD4 T cells as a way to kind of catch fluctuations in autoimmunity. Can we see this in human beings? So what our strategy was, we relied on as little as 20 mLs of blood, glycol, isolated PBMCs, stained with DQ8 tetramers that were had 4 different regions of insulin in them. So insulin 1220, 1321, and also 2 regions of peptide. And these were, I'll get into why we selected those regions. But they basically 2 arms for the human studies. The first arm is single cell sorting of tetramer positive cells, creation of cDNA library that has 96 T cell activation genes, as well as 48 TCR genes, which then allows us to pair gene expression profiling via the Fluidigm biomark platform with TCR signaling of those same cells. The second arm is that we took that sample and split it, and we put it through surface characterization by spectral flow cytometry. So I mentioned that we had a reason for why we picked insulin 1220 and 1321, and also connecting peptide, which is what CP stands for. The reason we selected those was because in that 2019 paper, what we found was in the islets of NOD mice, at 6 weeks of age, we could find mice that had a range of 1220 positive cells, and they were very low for 1321. But at 12 weeks of age, we actually found that the 1220 declined. This gave us confidence that 1220 specific, the CD4 T cells should be early on an activated cell type. The reason we picked connecting peptide was works by 2 papers from Stuart Mannering's group. First one here from 2015 was really interesting because they found connecting peptide specific CD4 T cells in the islets of humans, a deceased human. And then they also found it later on, a few years later, in the blood of people who were just became diabetic, but also one individual who was non-diabetic. So our human studies have 4 cohorts because we can't, we didn't have the ability to sample somebody over time, and we need to assess people in different stages of the disease. We had 4 cohorts. Our first cohort was a normal blood donor cohort, and that were people who are not at risk for type 1 diabetes, but they do have DQ8. They are DQ8 positive. Our second cohort are people who are from the TrialNet cohort, and these are individuals who are at risk for type 1. So they have a first-degree relative with type 1 diabetes. Our third cohort is a just diagnosed cohort. So these are children here in San Diego. We had a collaboration with Dr. Mike Gottschalk, and children who would come in just coming in as diagnosed with type 1 diabetes. We were able to sample blood from them. And our fourth cohort was a group of established type 1 diabetics here at UCSD, in collaboration with Dr. Jeremy Pettis. And these are people who have type 1 diabetes for decades. So I just want to emphasize that all individuals enrolled are DQ8 positive. One other feature that I want to mention is that we relied on just fresh blood. And the reason we did that was because we had done a couple of tests. And in this case, what I'm showing here is we took 2 individuals, we isolated their PBMCs, sorted antigen-specific cells, and that's here on the left here with fresh PBMCs. Then we froze the remaining PBMCs down, kept them in liquid nitrogen for about a month, thawed them out, and then resorted antigen-specific cells, so insulin 1220, 1321, and the 2 connecting peptides. And what we found here is that disproportionately, the activated cells were impacted by freezing. And we have a stark decrease in the number of activated CD4 T cells. So for this reason, we decided that we were only going to use exclusively fresh samples that were isolated fresh. So when we, as I mentioned, we profiled antigen-specific cells over 29 individuals by RNA, flow cytometry, and TCR sequencing. So let's start with the RNA, and then we'll move on to the next parts of that. So I'm just going to give away the point of the study of just are the findings, because it gets kind of complicated, but I'll just walk you through it. So we did an RNA assessment of 96 activation genes. We had approximately 4,000 insulin specific cells. We combined all the data across 29 individuals together and applied k-means clustering to the data. What we found was there's 2 large groups. There's a high expression group, which I'm going to refer to as high, low expression group, which will be referred to as low. We then subclustered each group, and we found that there were 3 high subclusters and 4 low subclusters. So what does the data look like? So this is all approximately 4,000 cells combined into a single heat map. And I think just visually, it's very easy to tell that there are 2 main groups. So this is the low group, which is defined by low given on the number of genes that they express, versus high, which express considerably more genes. One other distinction is that most of the genes that are common between these two are things like housekeeping genes, like GAPDH, RIPL13A, BCL2. What defined the high expression group, so these were genes that were unique to that group, were genes that were for activations like CD44, ICOS, NR4A1, NF-kappa B. Roughly the split is about 22% in the high and about 78% in the low. When we used UMAP to visualize this, what we found was, as we saw, a sort of a low expression and a high expression group. But what we didn't expect was to see these different lobes. And I'm mentioning that for a reason. We'll get back to that. When we overlay the distribution of the different clusters, what we find is, for the most part, there's reasonable intermixing between the different cohorts, with the exception here of the gray group, which is the normal blood donor, and I'll get back to that. So as a first pass assessment, what is the frequency of the distribution of all the insulin specific cells, but also individual specificities? So that's what we're depicting here. At the top, it's the frequency of total insulin specific cells in that high expression cluster. We're finding for the normal blood donor group, it's low, whereas there's a trend for an increase for people who are monitored, just diagnosed, and people who are established. This was pretty much the same trend for each of the individual specificities, where it was normal blood donor had the lowest percent in the high, and the just diagnosed had the highest percent in the high expression group, and the established had sort of like a mid-tier frequency. These results, there's a trend here. It didn't pass our Bonferroni corrected p-value, which means we would need more numbers, but the trend is clear that there is an increase as people become diabetic. So what I'm going to do now is focus in on that low expression group. And when we look at that group, what we're seeing is that, as I mentioned, there's 4 subclusters of low expression cells. So when we just subsample those or subcluster them, we find here is 4 groups, and that group that was enriched for the normal blood donor is called low 4, as I'm showing here in the UMAP. When we take a look and say, how do these antigen-specific cells distribute across all the low subclusters and the unified high expression cluster, we combine it by cohort, what we find is that the individuals in the normal blood donor cohort have the highest percent of their cells in that low 4 group. Now, that low 4 group, what defines it? So interestingly, what defines that group is they do have, again, more genes expressed than the other low subclusters, but the genes that they do express are really intriguing. They have NR4A1, so it's a gauge of TCR signaling, so meaning that they did receive, or suggest that they did receive TCR signaling for their antigen. They have interferon gamma receptor 1, and also highest expression for like TNF, JAK2, which suggests that they've seen their antigen, but they have incomplete activation, almost like a signature that it might be a type of a peripheral tolerance has been applied to them. This low 4 group declines in the monitored group and is not present in the just diagnosed or established. When we break it out by individual, what we're finding is it's pretty consistent for the individuals in the normal blood donor group. They are consistently have most of their cells in that low expression, so tolerized subcluster. When we break it out for the monitor, we find that many of the people in the monitor don't have their cells in that low expression group, with the exception of one, which is monitor MON15. And again, by individuals, we didn't see any variability at the individual level for people who are diabetic. Now let's transition to the cells in the high expression group. When we take a look at that data, what we find is there's 3 subclusters. And these subclusters are defined by, so cluster 1 is a Treg phenotype. It is expression of FOXP3, TFH group, which is expression of BCL6, and a T memory group. And this is sort of defined by sort of low expression of many activation genes that are very common across both the memory and the other two clusters. One thing I'll point out is the TFH group also had some of the highest expression for HLA-DR, which is an activation marker for humans. When we project out on the UMAP here, we again see that for the most part, they separate well, and there's some good intermixing between, it appears to be some good intermixing between the different cohorts. But when we apply the same analysis again, when we look at, and we find, and what we see here, so if we apply the same analysis where we look at all the antigen-specific cells in the high expression group and say, how do they distribute across the different high subclusters? What we again find is that for the normal blood donor, you get a higher percentage of those cells in that Treg phenotype group and a lower one in that memory compartment. But as you move across the bar graph and move towards diabetes, you see that there's a decrease in that Treg population, but an increase in that memory, and a small increase also in that TFH population. What you also find is that there's this Treg population that kind of sort of attempts to reestablish itself in the established individuals, which I think is pretty interesting because it suggests that there's sort of this ongoing tug and pull even in people who are established type 1 diabetics. One thing I would point out is the way this distribution was done are by statistical analysis, this is significantly different between the cohorts, and that was also true for the low expression group as well. Coming back though, when we look at how these individuals, when we break out the individuals from the individual cohorts, what we're finding is, for the most part, the normal blood donors, again, are pretty consistent. There's higher percentage of the cells in the Treg group and a lower percent in the memory. The monitored group is pretty interesting because about half of them have a phenotype or a signature that looks very similar to what you find perhaps in the diabetics, and the other half look somewhat similar to what you find in the normal blood donors. And there's some sort of a mixed signal from high too. Again, for JD, we see some consistency, but there's sort of this increase in half of the people with this TFH population. But again, if we look at the established, this is, I think, one of the more intriguing parts of the data, which is that, again, you have these two individuals and maybe a third who are reestablishing that Treg phenotype, which again suggests that this process is still active in people with established type 1. So moving on to TCR analysis. We were able to sequence 570 single TCRs, 180 of which are paired. And when we look at the different motifs of the TCRs that recognize different insulin regions, from the P9 model, we should expect to see a negative charge because 1220 is a peptide that does not have a negative charge in its 9th position. What we find is that actually in normal blood donors, we do find that negative charge here. And that we can still see it in some of the monitored and just diagnosed, but it's not as prominent, which does fit with that P9 model that the early people in early stage of autoimmunity are going to have that negative charge in their CDR3 beta. When we do it by percent, so we take the total TCRs that we sequence in each cohort and we say what percent have a negative charge in their CDR3 beta, we find that the normal blood donors have the highest percentage of TCRs with a negative charge, and this declines as type 1, the risk increases and onset occurs of type 1 diabetes, which supports that P9 model that we found in non-mice and through previous studies. So now let's transition to that second arm of the study, which was profiling antigen-specific cells from humans with spectral flow cytometry. And in doing so, this was our panel here. So we had our tetramer. So again, we had the same 4 specificities and a panel where we could assess activation through these different number of markers. Again, I'm going to give away the end of the story because it can get a bit complicated. Well, basically, we assessed almost 2,000 insulin-specific cells. We combined the data across different people, normalized it, applied a clustering algorithm. And what we found was that there were 4 main groups of cells. So looking at that data, here at the bottom is the UMAP of, or sorry, at the top is the UMAP of the insulin specific only cells and how they project on a UMAP. At the bottom is how those cells were situated in that larger CD4 population. So the point of the show is that they kind of spread out through the bulk CD4 population. And as I mentioned, we came up with 4 clusters. So T1 is a TH memory population. Is kind of based on the expression of 127, CXCR3, mid expression of 62L, and TIM3. For T2, we have high expression of ICOS, PD1, CD69, and CD28. T3, which is that regulatory phenotype, so that was very high CD25, low expression of 127. And then finally, our what we are calling a tolerized T4 group. So this was defined by LAG3 and CTLA4, so hallmarks of antigen experienced cells. When we do the same analysis again, where we take all those antigen-specific cells and we just show project them onto this sort of a bar plot, and we say what percent were in each one of these 4 groups by cohort, we found statistically significant differences by cohort. We found that there was more of a memory phenotype, at least at the surface protein marker level, for people normal blood donor group. A increase in the activated group here in blue for children who are just coming in as just diagnosed, but then also an increase in that regulatory population, which again suggests that there's that tension between tolerance and autoimmunity. And then again, we see an increase in that memory population for established type 1 diabetics, a reasonably activated population, which again suggests that this process is still ongoing, and a regulatory population that looks similar to the normal blood donor group. The T4 tolerized groups stayed pretty consistent across all 3 cohorts. So when we break out by individual here, what we're again finding is for the normal blood donor group, pretty consistent results. For the children just diagnosed, some degree of variability. So this particular person had a much bigger activated population, whereas the other 2 had more of a regulatory, higher percentage of cells in the regulatory group. But in established, again, we see some consistency here. But again, there's that regulatory population that's variable between them. So we also had that monitored group. So how did they look? When we profiled them by spectral flow, we found that there was some consistency with the first, maybe let's say 5 individuals, and then a little bit more variability in perhaps the last 5. We also applied support vector machine to try to predict where these individuals would sit. Because as we can see here, many of them have that bigger memory population, but this individual, 14, has a really strong signal coming from that regulatory group. So we asked, where would these sit relative to where the 2 diabetic groups as well as the normal blood donor group would sit? And when you apply SVM, what we find is these are the predictions we got out of it, which was, so N stands for normal, J for just diagnosed, E established. So this particular individual cells sat closest to someone who was part of the normal group. And that's kind of what it means by normal. When we overlay antibody status, what we find is the individuals who had a normal prediction generally had really only 1 autoantibody, though there was this individual who had 3. Whereas it was a little bit more of a spread for the individuals who were predicted to be closest to diabetic status. When we also overlay the OGTT, for the most part, people who had a normal prediction also had a normal OGTT with the exception of 1, whereas the individuals who had their prediction to be closest to being diabetic actually had either limited normal or not normal OGTTs. We tried to estimate the accuracy of our results, and we found that it comes out to be about a 70% accuracy with just 1 time point. What this shows is that, again, we can rely, it really lays the groundwork for that we can rely on these circulating antigen-specific cells as a way to kind of gauge where someone is in the autoimmune process. And it, in some ways, does follow the same trends as what we can find with the metrics we currently have. But see, we also had RNA data and protein. So our next question was, could we combine the 2 together to try to see if we can increase certainty of our prediction model. So to this, we turned to a different type of model, and what's called a Gaussian mixture model. And what we did here, so on the table on the left, we trained that model on our 3 known groups. So we had the normal blood donor group, the children, for just diagnosed children, and established type 1 diabetics, and we trained it, and it, when it had a pretty high certainty based off the data we inputted. When we then put the monitored group into it, we actually were quite pleased to see that the Gaussian mixture model actually improved prediction and certainty. It labeled most of the individuals as being diabetic, with the exception of MON15, which was that same individual back at the RNA data who had a very high percentage of their cells sitting in that low expression 4 group that I showed earlier. But what the main point of this is to say is that there are different models that we can use. We can combine RNA and protein data, and it can improve the prediction and certainty. And the final point I want to make is that we did have one individual that we were able to sample twice. And the reason that we were, this person was not in the main study. This is data, that we have the opportunity to present now. But when we sampled an individual at time point 0 and then 1 year later. And what we're showing is that low expression group here. At time point 1 in red, most of the cells sat in this low expression 2 group. But 1 year later, we found most of the cells actually sat down here in that low 4 group. And that's what this plot here is showing. What the point of, I think this is trying to make is that, again, individuals, the experience of moving through autoimmunity is very individualistic. And what you catch on one time point may not be representative. What I'm trying to say is, I guess, one time point isn't sufficient. We need to have more sampling of individuals because they probably are going through a relapsing remitting type of autoimmunity. So the conclusions of our study are that circulating insulin-specific CD4 T cells are a diverse population, and that's true at a protein and transcript level. At the transcript level, we have 2 groups, cells that have high expression of many activation genes, and a group that has low expression of those genes. And within those groups, there's diversity as well. At the protein level, we have 4 main populations that we've identified. The proportion of these anti-insulin CD4 T cells in each phenotype adjusts with the risk. So as we saw, individuals who are moving towards type 1, the way those cells spread across the different clusters changes, and that we did find was statistically significant. At a TCR level, we are able to find signatures of the P9 model. So that's that negative charge in the insulin 1220 specific CDR3 beta region, and this recedes with risk. Not finally, but I think a strong point for us was that we can differentiate normal blood donor cells from those at risk with type 1 diabetes, both at the RNA level and at the protein. I think what this suggests, all the data combined suggests, is that the autoimmune process is still active in people with established disease, and that for these individuals, perhaps intervention is still possible. It also suggests prediction of diabetes may be possible via anti-insulin CD4 T cells, and it may be a much more granular way, in an individualistic way, to gauge what an individual's experience of autoimmunity is. And the earlier we can diagnose somebody, the earlier we can gauge how fast they're moving through autoimmunity, the better and the easier it will be to intervene. We'll have moments where we can intervene and say, this person is accelerating through, we now have a mechanism to intervene, so let's do it. The future directions of the study are, we need to do longitudinal sampling. We'd like to have an expanded cohort of at least 100 individuals where you do multiple blood draws per year, and you're able to combine that and then retrospectively take a look and say, this person became diabetic. What were the features that happened preceding diabetes? What was the experience of an individual? We're going to fine tune our RNA and protein surface marker panels because we can always get better. And we really want to develop a prediction model utilizing both RNA and protein assessment and really push the accuracy above 90%. And we believe we could do that with longitudinal sampling. With that, I'd like to acknowledge quite a few people. It was a translational study. It was a collaborative study. It took a lot of people's help to get it done. First and foremost, I want to thank Luke for giving me the opportunity to do the study, everybody in our lab who really came through and helped out. Want to thank Shu Chin Tan, who really helped with the analysis, and the Sue Lab here at Scripps. I really have to thank our clinical collaborators. So just start with Jeremy Pettis here at UCSD and Adrian Armstrong for helping with the established group. Mike Gottschalk for really helping us with the Just Diagnosed. Peter Gottlieb and Kate Speak for TrialNet and really helping us, plus the TrialNet in general for even creating the cohort so that we can assess these individuals who are at risk. Thanks to the Genomics Core, our Computational Core, Flow Core, and I really like to thank the Sugar Science and Monica and the entire Sugar Science team. It's really great you guys created this community. I'm not particularly aware of any other groups that have this kind of place where we can meet, talk, and share science. So thank you. Happy to take questions.
Thank you so much for that. Wow, this is a beautiful talk. Congratulations on your new paper, very elegant.
Thank you.
I have a couple of questions, and if anyone else in the audience has a question, please put your hand up or just unmute yourself or drop something in the chat, and we can certainly address these questions. I know this talk was also very well received at IDS Paris because we were there, and it was just a lot of people were talking about it, had quite a bit of buzz. So I wanted to ask you a couple of things. Can you discuss the protocol around this sort of fresh human blood procurement, the timeline and the handling, and, just talk a little bit, how will you scale this from the, number that you have right now to this hope to gain 100 patients?
Yeah. So what we found was that blood can be shipped to us as long as it's kept, even at room temp or it's slightly below. Whole blood can be shipped to us, and we didn't find any increase in death of cells. So blood was shipped to us from Colorado, from Seattle, through our collaborators, San Francisco, and we didn't see any perturbation in the data at the RNA level or at the protein level. So 24-hour blood shipment across country we found works.
So that's sort of the shelf life is the 24-hour.
Yes, we've only looked at 24 hours. We didn't push past that, but 24 hours, the data looks very good, and intermix as well.
So you guys are working quickly over there.
Yeah,
that's so that's fantastic, and a tribute to your dedication to learning more in this piece. And then just in terms of the scaling, how might you get this sort of up to 100 patients right there in San Diego? Is it possible, or are you going to cast a wider net?
I think we're probably going to cast a wider net. We can definitely pull just from San Diego, but I think the strength would be able to show that this is something we can you sample people not just in San Diego, but different geographic environments as well.
Okay. I wondered, when you're talking about the decrease in Treg, the decrease in TFH and increase in memory cells as people move towards T1D, and you really kind of tapped on this ongoing tug and pull, even in patients with established T1D. Can you talk a little bit about this sort of signature in other diseases where that's true? Has it been noticed, or is this sort of like the first wading into that? People who have established autoimmunity, do you know of any other papers or other people looking at this?
That's a good question. I at the moment can't recall another one. Perhaps there has been. I think one thing I can think of is, there's different studies for like different bouts of inflammation and like relapsing, remitting, but sort of that tug and pull. Yeah, it's unfortunate at the moment. I'm kind of blanking.
Yeah, it just feels to me that this is something novel that you guys have really unearthed. So I wanted to kind of just highlight that. And then I also wondered, do you think that the T1D TCR BCR data repository that we're building together with our partners at iReceptors and sort of this federated group of scientists across the US and Europe will inform and illustrate the phenotype of the sort of prodrome you're seeing? Do you think that these existing samples that people have run through adaptive, et cetera, can also illustrate what's happening in the immune, prodrome, or is what you have found here, could this be a missing signature that also has to be included?
So I do think the combining of different TCR data from different groups can be a great way to kind of look at the disease, and as you're saying, I think I think we should also have sort of these antigen specific cells and sort of the bulk population together, and try to like project from the antigen specific onto the bulk and see if you can find those signatures. I think having both arms is really going to be beneficial.
Big picture, and it's almost like rough and fine tuning.
Exactly, yeah. So you want to have that sort of like big picture capture an image, and then also these sort of individual images, and put them all together. I think that there is going to be value in that.
Yeah. We hope so. And then I guess, the last one is, how might this autoimmune process, being still active in established diseases, intersect with this sort of senescence hypothesis or senescence work that's coming from, Peter Thompson's lab and others? The beta cell senescence, like, okay, maybe, there's sort of bouts of autoimmunity when senescence occurs in these beta cells. And then there's a possibility they could come back online if you could just unlock what's happening.
I think it probably lends credence to it, but I would also say that, one thing we talk about in the lab a lot is that, type 1 isn't this like massive culling of islets, and the islets are spread throughout the entire length of the pancreas.
Yeah.
And it's almost as if type 1 is sort of this elimination of individual islets over time. And so maybe these bouts of autoimmunity, or you have this sort of burst of you're losing a few at a time, or maybe 10, 20 at a time, hypothetically, and then it goes quiet because the immune system hasn't seen the other islets. So perhaps it's senescence, perhaps it's the immune system has yet to see the islets, but I think the point still is, there's still beta cells that can potentially be saved for use for more natural control of glycemia.
Yeah. Well, I've asked so many questions because I just found this talk to be so interesting. Anyone else? Unmute yourself, or you can reach out privately to Siddhartha Sharma and the Luke Titan, and at the Titan Lab at Scripps in San Diego, doing some really amazing work. It's beautiful. And it's really, I cannot wait to see how it goes when you scale it up.
Thank you. We're looking forward to it.
Thank you so much for joining us. Appreciate it. And we'll speak again soon. Bye-bye.
Thank you, Monica.
Have a good day.
Thank you.
Bye.
Thank you. Bye-bye.